Hi everybody, and welcome to the latest VodCast for Honors Biology at Desert Ridge High School. I'm Mr. Galladay, and today we are going to be talking about sex-linked traits. Sex-linked traits are a pattern of inheritance that is different from the Mendelian pattern of inheritance. We've already talked about several others, and this is a good point to update your table of contents in your notebook. And here we go. We're going to start off with a little genetics problem, with a Punnett square problem. And to begin with, we're going to look at this problem that involves fruit flies. We're going to look at these two fruit flies, one of which is a female and has white eyes, the other of which is a male and has red eyes. And in this particular trait in fruit flies, the red eye color is dominant and the white eye color is recessive. P1, so in other words, both of these fruit flies were born from true breeding parents. Okay, so that gives us a little bit of information. And the thing that we want to know is, what are the expected phenotypic ratios for the F1 and F2 generations? So you say, Mr. Galladay, this is easy peasy. I could do this with my eyes closed. So we start off, we need to, of course, know the genotypes of the parents. And since they came from true breeding parents, it's reasonable to assume that the female, since she has white eyes, would have little e, right? White eyes are recessive. The male, we would assume, came from true breeding parents, so he has big E, big E. That's his genotype. So the next step is to find out what gametes they can produce. So we simulate meiosis at this point. So the male can only produce big E gametes, the female can only produce little e gametes, and so 100% of their offspring would be heterozygous, and the phenotypic ratio would be 100% red eyes for the F1 generation. For the F2 generation, we would take two of these individuals, and they are both heterozygous. So so there is the genotypes of the parents. When they produce gametes, both of them will produce either a big E or a little e gamete. So when fertilization happens, we have 25% are homozygous dominant, 25% are homozygous recessive, and 50% are heterozygous. So that would give us a phenotypic ratio of 75% red eye and 25% white eyes. And you're going to look at that and say, that's perfect, Mr. Galladay. I came up with that. Where's my cookie? And I'm going to say, hold on just a minute. Because what we're going to do next is get some actual fruit flies and get some actual data. So here we have our handsome red-eyed male and our lovely white-eyed female, and we're going to have them produce some offspring and see if the data fit our model. So they produce almost 2,000 eggs. Wow, a lot of kids. Imagine the number of shoes they're going to buy, because each one's got 6 legs. Anyway, when we look at the kids, 913 of them have white eyes. 894 of them have red eyes. Okay, now that does not fit our expected phenotypic ratio. We would expect to have 100% of them have red eyes. Something's wrong. Now when we look at the F2 generation, what do we see? Well, we see again about a 50-50 split. We have about 1,000 of them have white eyes and about 1,000 of them have red eyes. Something doesn't fit. Let's look at our data a little closer. When we look at this a little closer, we see something else that is very strange. Look at this. Of those 913 with white eyes, all of them are males and none of them are females. Very strange. And of the ones with red eyes, they are all females. None of the ones with red eyes are males. Okay? So we might think, well, we've got a pattern that only males can have white eyes. Well, but that's not true, because our father had red eyes, and now also when we look at the next generation, we see that it splits pretty much 50-50. So you might think, well, this is just craziness. This doesn't make any sense. And you're right, it doesn't make sense with the standard Mendelian pattern of inheritance. It does, but there's something else going on here. And what is that other thing that's going on? Well, that's what we're going to find out. This problem was first addressed by this guy. He's Thomas Hunt Morgan, and he worked in the early 1900s. And he bred thousands and thousands and thousands of fruit flies. And you see these vials all have thousands of fruit flies in them. Here's another picture of his lab, and you see this young lab assistant, and he is surrounded by all these vials which all contain thousands of fruit flies. This is a close-up of a more recent picture. Drosophila melanogaster, which are the fruit flies, these are still the workhorses of genetics. We still use them today in lots of genetic experiments. Their genome has been completely mapped. And so here you see a close-up of what these are mostly the pupa, which are kind of like the cocoons of the fruit flies, and you see a couple of adults walking around in there. Okay, so what these are is these are sex-linked traits. Genes are on the X chromosome. We're going to come to the answer of this problem a little bit later on, but for right now, let's just see what sex-linked traits are. These are genes that are on the X chromosome. Now, sex-linked traits can also be on the Y chromosome, but because the X chromosome has many more genes, they are almost always conditions that are on the X chromosome. Since males pass their X chromosome to their daughters and their Y chromosomes to their sons, this causes a very different, you see, different patterns of inheritance depending on the gender of the offspring. Now, females always pass one X chromosome to all of her offspring. Females with one recessive gene on an X chromosome are called carriers. Okay? Males always inherit one X chromosome from their moms, but males only have one X chromosome. So that means that they are much more likely than females to inherit any recessive disorder. Some common human examples are red-green color blindness and hemophilia, and we're going to talk about both of those here and see what those are. Okay, so you may have heard of color blindness. You may have color blindness. One way to tell is to take a test like this. This is a circle, obviously, with lots of colored circles in there, and you probably see that there is a pattern in there. You may see a number 74, you may see a number 71, depending on your color acuity, or you may just see a bunch of circles, and you can't really see any pattern at all, or it may be very, very faint. This is a standard test that is used to detect color blindness. And one of the more common forms of color blindness is an inherited red-green color blindness. It is a recessive condition. It affects about 8% of males and about 0.5% of females worldwide. The reason for the differences we will see very shortly as we go through this. So here we have a female that has two X chromosomes. The normal allele is a large B, right? And the colorblind allele is a small b. So here is a female that would be a carrier because she's heterozygous for the condition. And here's a male who would be colorblind because he only has the colorblind gene on the Y chromosome. He doesn't have anything. So he only has one gene which affects that particular part of his vision. So he inherits that particular disease. So females can either be normal, which would be big B, big B. They can be carriers, which would be big B, little b. Or they can be colorblind, which is the homozygous recessive condition. Males can be normal, which would have a big B and a Y. Colorblind would be a little b and a Y. Notice that the males only have one gene because they only have one X chromosome. The Y, even though it's homologous with the X, it doesn't have the same genes on it. Okay? Let's look at a couple of problems dealing with this. And here's the first one we'll look at. And so here's a female and a male. And the question is this: can this couple produce a colorblind son? Okay, well, if we look at this, we notice that the male, the father here, is colorblind, right? And we notice that the mom has normal alleles, so she's a big B, big B. So both of her X chromosomes have the normal the normal allele. So you might at first suspect that, well, of course he can, because the father is colorblind, so of course he can produce a colorblind son. But wait a minute. What is it that makes his son a son? And the fact is that all of his sons do not inherit this chromosome. They inherit the Y chromosome. If they inherited an X chromosome from this man, they wouldn't be a son. They would be a daughter. So there is actually no way that this couple can produce a colorblind son, because no matter what, the X chromosome that the son inherits is going to come from the mom. The Y chromosome will always come from the dad. So what does this mean? Well, all the kids were going to be phenotypically normal. That means that they're going to always get one good allele from mom, so none of them will be colorblind because the condition is recessive. All of the daughters will be carriers, which is to say heterozygous, because no matter what, they're going to get one good chromosome from mom, and they're going to get one chromosome, the X chromosome from dad, will have the colorblind gene. And no matter what, all of the sons will be normal, which is to say they will be big B with a Y chromosome. Okay, here's another problem which is very similar, and the question is this: can this couple produce a colorblind daughter? So what do we have? Well, we have a mom who has one normal allele, which is big B, and one colorblind allele. So she's heterozygous, which would be big B, little b, and then the dad has a single good chromosome, so he would be a big B with the Y chromosome. Okay. Well, you might think again that because the mom has one bad chromosome, you might think, well, yeah, they could inherit that from the mom. But in fact, all of their daughters will be phenotypically normal, again, because no matter what, they're going to get one good X chromosome from the dad, right? So dad they're always going to get one of their X chromosomes from dad. If they were to get the Y chromosome from dad, they wouldn't be a daughter, they would be a son. And so no matter what, they're going to get this chromosome from dad, and they have a 50-50 chance of inheriting the bad chromosome from mom. So they have a 50-50 chance of being a carrier. Okay, the sons have a 50-50 chance of being colorblind. Why? Because the mom, they're going to get their X chromosome from mom, and they have a 50-50 chance of getting an X chromosome that has the colorblind gene on it. No matter what, they're going to get a Y chromosome from dad, which is why they are sons and not daughters. Okay, so the next trait, or the next condition that we're going to look at, is called hemophilia. This is also a sex-linked condition. Let's talk a little bit about what hemophilia is before we get into the genetics. The normal condition is this: anytime you get a cut or a bruise, a blood vessel is broken or cut open, there is a protein in your blood which is called fibrinogen, and this is contained in the platelets, which are part of your blood. It reacts with the air to form a clot or a scab, which stops the bleeding. A person with hemophilia has a set of instructions for that particular protein, which is to say the gene, and the instructions for making fibrinogen is faulty. In other words, it makes fibrinogen that doesn't work, doesn't form a clot, and doesn't form a scab. Now, the fibrinogen gene, as we've said, is on the X chromosome, which is why it's a sex-linked trait. So here's a question: how many copies of the fibrinogen gene do females have? Well, of course they have 2, because they have 2 X chromosomes. And how many copies of the fibrinogen gene do males have? Well, of course they only have 1, because they only have 1 X chromosome. Males have 1 X and 1 Y chromosome. Here's a little diagram to show how this works. Our blood, as you know, contains red blood cells, and it also contains these little things that are called platelets. And the fibrinogen is a protein which makes up the platelets. When the blood comes in contact with the air because of an open wound, the platelets sort of, well, they don't really explode, but they kind of fall apart or break apart into this sort of mass of sort of like a very sticky spider web, and that's produced by the fibrinogen. So again, this happens to a person that does not have hemophilia and has a good fibrinogen gene. And so under a microscope, this is what this looks like. And so you see all of the fibrin, which is produced from the fibrinogen protein, and the fibrin proteins form this kind of like a spider web, which traps a lot of blood cells and basically forms a scab and stops the bleeding. In order to study this, we're going to look at we're going to just do a real quick investigation into pedigrees. Pedigrees are a tool for tracking trait through several generations. You don't have to copy all this stuff down, but I just want to let you know real quick what a pedigree looks like. Females in a pedigree chart are shown as circles. If they are if they have a particular condition, they're shown as a filled-in circle. If they're a carrier, which means they're heterozygous, they would look like that, and unaffected females are an open circle. Males are squares. That's not that doesn't mean anything disparaging against males, but on a pedigree chart they're shown as squares. A darkened square means the male has the condition. A light square means he does not have the condition. Couples that are married are joined by a horizontal line, and their offspring are a vertical line coming below that. So this would be a husband, a male and a female that are both normal, that don't have, in this case, hemophilia, and they have a male and a female, a son and a daughter, that are shown like that. Okay, so we're going to look at a very famous case of hemophilia. This rather regal-looking lady is Queen Victoria, who ruled in England for most of the 1800s, and she was very famous as a carrier of hemophilia. And since many of the royalty in Europe intermarry, her offspring, and they're fairly famous people, their offspring have been traced. So her husband was Albert, and there she is. She is a carrier of hemophilia, and these are all of the children and the spouses that they had. So here is the line which became the current. This is their oldest son, Edward, who became King of England, and his offspring are the parents of current Queen Elizabeth. And as you can see, they had one daughter who was a carrier, one son who was hemophiliac, and another daughter, Beatrice, who was also a carrier. As time went on, they produced many grandchildren, some of whom intermarried. Here you have a case of some cousins. These are some first cousins that married. Henry of Prussia, not a good choice because his wife Irene was a carrier, and so you see here several grandchildren which have hemophilia, and so you can see over the generations this condition just passing from generation to generation, and the current. Okay, so here's the problem that we started off with at the beginning of this. So here is our white-eyed female and red-eyed male. And as we said before, the red eye color is dominant and the white eye color, little e, is recessive. But here's the thing, the eye color genes are on the X chromosome. All right, so what I'd like for you to do on the left-hand side of the page is to draw Punnett squares that correctly predict the observed F1 and F2 phenotypic ratios. Well, there's our observed results. For the F1 generation, we have of the offspring from the F1 generation, 50% of them are males, and they have all of the males have white eyes. 50% of the offspring are females, and they all have red eyes. For the F2 generation, we have 50% white eye, 50% red eye, and 50% are males and 50% are females. So the red and white eyes, as well as the gender, are pretty evenly distributed. Remember, it is not our normal dominant and recessive kind of pattern because of the fact that the eye color gene is on the X chromosome. So the males will have either a big E and a Y or a little e and a Y. The females will all have two genes as normal. Okay, so do this on the left-hand side of your notebook, and this is going to conclude this podcast for Honors Biology at Desert Ridge High School. I'm Mr. Galladay, and I hope you have a great day.